Good morning, everyone, and thank you for joining us for this third day of Varsity Sci. And today we will have talks on the subject of scientific computing. We will start off with Loïc, who will tell us more about his work on green algorithms and quantifying the carbon impact of computations. So how much environmental impact a computational work has. So without much further ado, I'll pass on to Loïc, our first speaker of today.
Thank you. Let me share my screen. All good?
Yep, brilliant.
Yeah. All right. Well, thank you for organizing such a great event. It's brilliant to have such a great lineup. And it's good to have an opportunity to talk about a topic that's a bit different. We're not so much talking about the methods or how to do it, but what's the impact of our work. So, well, unfortunately, there are lots of examples of the consequences of climate change. But this project started following the terrible Australian bushfires last January. Well, the pictures are striking, and we realized that really we couldn't really ignore the health burden of all these events. And we had to do more to address the carbon impact of our work. So more and more large-scale computations, they are done in large data centers. It can be the institutional servers like Cambridge and Oxford, they both have theirs, or it can be cloud facilities like Google Cloud. But the first question is, well, whether data centers do have a large carbon impact. Well, that's not a surprise that the answer is yes. And we could pull a lot of different numbers to show that, but there's one that I find particularly striking. Data centers around the world, they emit 100 megatons of carbon dioxide every year. For context, that is as much as the entire American commercial aviation. Of course, a lot of that is not related to scientific computing at all. But, well, I do spend a lot of time on running models and training machine learning stuff. So I really can't help but think that we need to take a closer look at our own impact as a community. So many modern breakthroughs have been enabled by supercomputers, well, and algorithms. Just for the fun of it, it was quite interesting to look at the most cited scientific papers of all time. Because it's a lot of biology this week, so let's start with that. The large representation of bioinformatic tools shows how widely used complex algorithms are in this field. That's insane, the number of citations there. Of course, it's not only biology, we can look at physics a little bit as well. They have these commonly used tools. But honestly, virtually every single field of science is concerned by that. And if we just look at this citation count and we think that even remotely it correlates with how much these tools are used, because that's usually the case, we're looking at thousands of tons of CO2 released in the atmosphere just as a result of research projects. Well, of course it's worth it. I'm not saying it's not worth it, far from it, but simply that like the financial cost of a project that would be acknowledged and planned for, and labs would be quite careful about that. The carbon cost should be similarly acknowledged and taken into account. So we worked on this with Jason Creeley here, a PhD student at the Baker Institute in Melbourne, and Michael Inouye. And we quickly realized that despite some initial work on deep learning, this entire aspect of computational research has been ignored so far. So we start small. So we start with just a blog article on the AGR UK website, mainly to raise awareness around that topic. And something that's interesting is that in today's data-driven world, a problem doesn't really exist until we can have data about that. So that's where we started. So our solution came as an open source online calculator. I'll talk more about this calculator in a moment, but let's start by just talking briefly about how you can estimate the carbon impact of an algorithm. For a long time, actually, people discussing environmental impact, they were like, oh, but we can't measure that, so let's just not do it. But actually we can. And to not have the talk run for too long today, I won't get into great details, but if you're interested, our preprint about the methods is out, so you can check it out on arXiv. So first, we need some metrics to quantify the impact. So most emissions, there are a mix of greenhouse gases like methane or carbon dioxide. So what we want is just one unit that will measure the global warming impact. Well, there is one quite widely used called CO2 equivalent. So that's great. We can use that. That's just one number that tells you what's the equivalent impact of what you're doing. And we need some context because I have no idea what 1 kilograms of CO2 equivalent represents. And you can look at like driving, and we do that, looking at driving or flying or this kind of thing, and it's great. But we ended up creating a new unit actually to answer the question. Well, actually, if you want to make your emissions disappear, sort of, how long will it take a tree to absorb these emissions? And well, we called that a tree month, which is just the the number of months it would take a tree to absorb a certain quantity of carbon dioxide. And obviously it depends on a lot of parameters, but we ended up with an estimate of around 11.4 kilograms per year, which actually conveniently, just to have an easy number in mind, it roughly translates to 1 kilogram a month, which is easy to remember. So, but we said that there are other ways to like bring some context. So you can look at driving. It's like 175 grams per kilometer, and that's like for a European car. American cars are a lot worse. It's like something around like 250. Planes and rail is actually really hard. It depends so much on how far you travel, mainly in the different types of lines. But planes can be anywhere between 130 and 250. And the rail is, well, it's not a surprise, it's the best one, between 6 and 41. Interestingly enough, the most efficient one is the Eurostar. And we usually don't think about it, but a search on Google has a carbon footprint as well. Not big, but well, it depends how many searches you do. And so does Netflix. And obviously these are like, especially Netflix, like with the more and more like high resolution and high definition thing, it will be a lot bigger and bigger impact. All right, so it's actually quite straightforward. To estimate the footprint of a model, we need two things. First, we need to know how much energy is drawn by the computer to run the entire thing. And that only depends on how efficient your hardware and your software are. But then you're interested in how is this energy produced, and what's the carbon impact of producing such energy. And that will depend on the geographic location and the energy mix of the country. So let's focus on the first part. I won't bore you with too many details, but just so you kind of have an idea of where it's coming from. It's relatively straightforward, actually. It depends on obviously how long the model is, how long your algorithm is running for. And then all the kind of hardware bits, so how many processing cores you have and how much power each processing core is using, and then how much you're actually using the processing cores. So that's like the first three. And then, well, the memory is drawing some power as well. So, well, how much memory is available and kind of the consumption of the memory. So you put all that together and that gives you some numbers. And then there's the efficiency of the data center. That the PUE, so power usage efficiency, it just measures how much extra energy is needed. So, for cooling or lighting of the facilities and this kind of stuff. And the more efficient your data center is, the less energy you waste on cooling and things like that. And that's what big cloud facilities like Google or Amazon spend a lot of energy and research on to optimize the data centers as much as possible. All right. Actually, memory is quite an interesting one. because you would think that the power drawn from memory is a bit like the processors. It depends on how much memory is available, of course, but also how much memory is actually used here, and how often you do access the memory to transfer stuff around. And some people looked at it, and actually, no, it only depends on how much memory is available, which is a great news because it makes estimating it a lot easier. The downside is when you request more memory just to be safe, like on a server or with like your research group. Well, I mean, I'm definitely guilty of that. Sometimes you just, oh, it might crash if I have like 12 gigabytes, so let's go for 16. Well, actually, you're increasing your carbon impact when you do that. So just something to keep in mind. All right, so we kind of cleared that bit. So now, what about the carbon? So what's the carbon intensity? So what's the carbon impact of producing all this energy that we've just seen we need? It varies greatly between countries, actually. And so actually, if you, for example, working on the cloud or have any flexibility in choosing your data centers because maybe like you're affiliated to several institutes or anything, that's the best way you can reduce your carbon impact, because we've seen it's just like a multiplicator. So if you multiply, if you're in a country that's like 10 times more efficient, then you will just divide your carbon impact by 10. That's very straightforward. But so when you look at that, running the exact same tool, exact same stuff, same thing in Australia compared to Norway, it's 80 times more, 81 times worse for the environment. So for example, this list is like the not exhaustive, but it's like all the countries we've curated and we've included in the app already. If you want more real-time data, you can, for example, go online and check electricitymap.org, which is great. Not all countries, and like the map changes all the time. So like, for example, here you can see that we don't have data on Germany, but actually maybe now there is data. It just depends on what time of day. But it's a real like hour by hour measure, and you can see what the different sources of energy are. So it's great to just like play around, and you can see that that's kind of crazy how bad Australia is, mainly because they're purely powered by coal. Yeah, so now we've kind of cleared that up, and that makes we have this formula, and we can just like plug in the numbers, and it gives us the carbon footprint of it. So of course, what we need is a tool to do it, because, well, this formula is nice, but we can't expect every researcher to kind of do the math by themselves and go try to find the value. I mean, it's just not reasonable. So we wanted to make it easy for everyone to do it, because we kind of said, well, we did the work once, maybe it's good if like other people don't have to do it now. So we have this online calculator. You just plug in basic information. You say, oh, my model was running for like 3 hours. Is like five CPU cores, well, 12 here, or 64 gigabytes of memory. Then you can say if you have like a local server, like something in your research group, or like an Oxford one or Cambridge one, or if you have, you can pick cloud, or you can say also if you're doing it on your local laptop or your desktop, that work, like you can do exactly the same way, of course. And then you just have to say like what kind of hardware you have. It's pretty straightforward. Then if you have extra information, then you can plug it in, and if you don't, that's fine. We just use like a default value. And when it just automatically adjusts here, so you have the carbon impact and the energy needed, and it gives you some context, how long it would take a tree to absorb it, and then the equivalence in terms of driving and flying. So yeah, what I was saying, when I talk about data center, it's kind of a general word, but a local laptop is kind of a small, tiny data center in itself. So if we scroll down, there's like kind of more details, just what's CO2 equivalent, what is a 3 month, some tips on what you can do about it. And then there's like just the details about the methods, and that's like a text you can just copy and paste. If you want to report it, you can just say, well, that just generated based on the numbers. But yeah, that's just an example of how you can be transparent about your emissions. it's actually quite fun. It even works on like phones and tablets for people who are so inclined. All right, so just if we summarize that, hopefully the app is easy to use. If you find it not being easy, just message us and let us know how we can make it better. So it's available online. The big thing, we didn't want to have people to like ask people to download things because, well, I hate downloading things that will like break all my pipeline and everything. So online is much better, and it's like completely free and open source. Like that was a big thing. We wanted to have everything online, so people, you can go and check all the data we're using and all the formula. You have even the code for the app itself. Like if you don't really trust the numbers or anything, or if you find bugs, please let us know. So yeah, all the data is available on GitHub. You can just go there, check out open issues. Like for example, if you want to have more countries and countries we didn't look at yet, or like hardware we didn't look at, there's like issues open. You can just like add a comment there, and we'll try to get to that quickly. Then there's just a quick question, which is how many times do you really run an analysis? And all these numbers we've looked at, I mean, and we will look at now, it's just for one run, but actually, I mean, you usually debug and it will like crash a few times, and then we try again, or maybe we will have to optimize some parameters, especially like, for example, in machine learning, it will be like hundreds or even thousands of iterations just to get one model we're happy with. And so that just multiply the impact. So we formalized kind of this idea through a pragmatic scaling factor. I won't have time to talk more about it today, but we've detailed it more in the preprint, so feel free to check it out or ask questions at the end. So then, well, now we finally have a tool. We didn't expect it to take so long, but now we had a tool. So we will let's look at science. So we kind of we wanted to have an overview of different fields of science. So we looked at physics, weather forecasting, some machine learning and natural language processing, and quite a lot at bioinformatics and genomics, just because we had the expertise in the lab. So let's forget about biology for a minute. We'll get back to that in a moment. So yeah, for particle physics, we looked at we basically for all that we use like published benchmarks, and we pulled the information about hardware and software and running times and things like that, and then we estimated the carbon impact. So it's not perfect, but it's kind of the best we can do just using published benchmarks. So we looked at DNA irradiation simulations using GAN4, weather forecasting. We look at IFS used by the European Center. And ICON, that's run for weather forecasting by the German Center. And for natural language processing, for people who are familiar with that, we looked at BERT, which is a very popular pre-trained model. So it's like a huge thing, huge model that's used everywhere in natural language processing now. And the numbers are pretty bad. So looking at like DNA irradiation, for example, when you include like the number of runs necessary to try different settings and different energy levels, it will take like 48 years. So 48 years to just absorb this one, like this experiment. And it doesn't get any better when you look at the other ones. The NLP one is kind of a mind-blowing 6,600 years to absorb this experiment, which is yeah, crazy when you think about it. So that definitely shows that there is a huge impact of science, and all that are great tools, and like of course we need weather forecasting and like simulating DNA irradiation is great, and maybe even NLP, but that we still need to acknowledge what the impact of it is, and just be aware that just be more transparent about it. Basically, that's all we're saying. And if we look at biology, so for all of you who are interested, so the results are not quite out yet, but there's a preprint that should be released soon, led by Jason Greeley, with great support from all these people. And we wanted to have an overview of like the carbon impact of what's popular in computational biology. So we looked at genome assembly, reads alignment, protein docking, molecular dynamic simulation. So we looked a bit at everything and tried to get like real world use cases. If we zoom out, we can even fit GWAS in the frame. So yeah, GWAS is not great for the environment. So I'll go quickly there because, well, we can discuss it more in details. But yeah, so basically that's just the details about the different tools we're using. We try to use like real world use cases. Not all of them are bad. Like some like RNA reads alignment, for example, they have like, it has really little impact, and that's great. Like that's part of the transparency of it. And what's really interesting is you can see, for example, for GWAS, if you look at the two first ones here, this is just updating from the first version to the second version of Bolt LMM, the GWAS tool, and you save like so much. Like the carbon saved is huge, and a lot of cases is about picking the best tool for the task. Same thing here, like LIMX versus TensorQTL, and some people will usually not consider it until they can't use LIMX, for example, for like running time reasons. But actually, just thinking about the carbon impact is kind of an interesting way to look at how to pick a tool as well. So of course, well, it shouldn't be the only criteria, but if it's one of the criteria picked and presented in the benchmark, that'd be, I think, a great win. But all the results presented today, actually, the orders of magnitudes are more important than the actual values, because well, each analysis, is a bit different. You don't exactly have the same like data, or you don't use exactly the same parameters or the same hardware. And we use published benchmarks, so we didn't run the jobs ourselves. It would be a bit ironic to like waste all this carbon just to publish a paper to say people to be more mindful of their carbon emissions. So instead, we use published benchmark, but we don't always have all the information we need. So the best way and the most accurate way to estimate the real impact is for the authors, you, to like run the models directly on your analysis. So this is, for example, what the team behind the Regini, which is also a GWAS tool, did, and they run their models and they try to present the results. Well, apparently Twitter thought the carbon impact was too shocking to be displayed, but once you accept that, they showed like a huge reduction in emissions compared to SAGE, which is another popular GWAS tool. So I'd say that if there's just one thing to remember from this talk, is that you can all like lead the way in your fields, especially like most of you being students. Like we're usually the ones running the analysis, and kind of, if we want to include that in our method, we usually can. So we have like a huge opportunity to be transparent about that, and just being a bit, more mindful of it. And like, some people even like use that and then get an estimate and then sponsored some like tree planting to offset their emission. And they were like, well, it will take, I don't know, 10, I think they measured their emission to be 3 years. And they were like, well, let's plant 30 trees, and that will kind of take care of that. So that's an interesting way to look at it, and that's great initiative. But if you want to remember a bit more about that, you can. These are the few like action points you can actually implement. So optimize your algorithms, or use the optimized algorithms if you're not writing them. Most of the time you will consider that when you are limited by like running times or memory usage, but actually, just consider optimized algorithm. That's obvious, but still. So limit the number of runs to a minimum. That means debugging on a really small scale, really small data set, so you don't run the model to see it crash after like a full night, which happened to all of us. And if you are doing parameters optimization, either in machine learning or something else, just keeping it to a minimum. Will it really make a difference if you optimize this parameter from 0.001 to 0.002? Sometimes it's tempting because you're like, oh, I can be a bit better and it will look better if I try to publish it. But still, it's just balancing the impact of running an extra 100 simulations. If you can, carefully choose the location of your data centers. As we saw, that's kind of the most impactful way to reduce emissions, is to go to greenest greener areas. And if you have any influence on that, you can even like push your data center to be more transparent about its efficiency, and why not be more energy efficient. You can offset your carbon emissions, as we've seen, by sponsoring like environmentally friendly projects. And mainly it's all about being transparent and report the emissions. The more people talk about it and the more people acknowledge their own impact, the more standard this practice will be. And I think that's definitely something that's quite important. All right, so to conclude, I'd like to thank all our collaborators who helped us along the way, both in Cambridge and Melbourne, and all the different funding bodies. And well, if you have questions, suggestions on how to improve the tools, or would like some new features, feel free to reach out. You can at these addresses, and I usually post regular updates about new features of the tool on Twitter, and alongside short summaries of what we discussed today. So thank you for your attention, and I'm looking forward to taking your questions.
Thank you, Loïc, for your very fascinating talk. So yes, I'm gathering questions from both YouTube and Zoom. If you're on Zoom, you could ask your questions in person or send them to me personally or to the chat. So I have one question. That's one question on Zoom. Are you working with research institutes to encourage them to use green algorithms?
Yes, so we've started to do that, starting with our own, but definitely that's the direction. Now that we have a working tool, that's the direction we're going to. And but for that, we usually, like, research institutes are quite protective of that, so we usually need to have a researcher who is part of who is already using the tool from the inside. And if a researcher is like, oh, I'd like to do that, so anyone who is keen to do that and would like to have their their specific hardware put in the tool, we really want to do that. So just get in touch, and we'll see how to set this up.
Thank you. There's one more question. You mentioned that the memory power draw only depends on the size of memory available. Does this apply to mobile phones and devices that we use in our daily lives?
Yeah, well, I wouldn't talk about. I'm not quite sure about like mobile phones, for example, and tablets, because I know they work quite differently, and I'm not an expert in that, so I don't want to like go there too much. And we haven't really looked at that because most of computations are obviously not run on phones. But for most of the standard memory used in data centers, it seems to be still the case that, well, basically the reason for that is these memory sticks are optimized for speed of access. So to make sure you can access anything really quickly, it kind of spread your data load over the space available, because if you have just everything at one place, it takes time to kind of drill through it. Or like if everything is spread along, it's really easy to access everything and to kind of see everything at the same time. So that's how that's how it's been set up, and that's why it kind of keeps all the memory awake all the time, and that's where the power consumption is coming from. It's possible that mobile phones, where the power consumption is a real problem, have been designed differently, and I'm quite hopeful that in the future data centers will be using more and more architectures that actually are mindful of power usage. So hopefully this will be changing soon, but yeah.
Thank you. There's one more question on Zoom from actually the next speaker. So Gita said, thanks, Loïc, great talk. Can you tell us the criteria that are best for identifying useful individual SNPs in GWAS data? So SNPs as in single nucleotide polymorphism.
I'm not sure. Do you mean for the benchmark we used? Well, our benchmark was using just Bolt LMM, so it was kind of a standard benchmark. It was running on UK Biobank using one trait, which was, I think, level of education, and just running the Bolt LMM model, or the way it's implemented. I don't know if that's answered the question.
Yes, you did. Thanks, Loïc.
Right. So I guess if there's still other questions, we will leave that until after the next talk. So thank you once again, Loïc. I'll now introduce our second speaker, Gita. So Gita is a PhD student at the Department of Plant Sciences at Cambridge. She will be presenting some algorithms that her group has developed over the past decade using simple coding methods and deploying this to make scientific breakthroughs in areas like digital systems, extinctions, and deep learning language. Without further ado, I'll now welcome Gita to deliver the talk.
Thank you, E. First of all, I'd like to just say, I'd like to just apologize for being late here. The internet connections at my end are really low today for some reason. I could not hear your introduction clearly, and therefore I'd like to ask everyone, you particularly, can you hear me? Can you hear me clearly?
Yep.
Yeah.
Yep, we can.
Yeah. If it helps, I can switch off my video just to make sure that the audio doesn't hamper. Can you still hear me clearly, everybody?
We can, yes.
Great. I'm going to share my screen here. Can everyone see my screen?
Yep, thank you.
Thank you so much. So once again, I thank E and everybody at Varsity Sci for starting out this series of talks for all of us to get to know each other better. And I really appreciate that you're also streaming it live on YouTube. I was not aware of that earlier. Thank you very much. First of all, I'd like to just fix a small glitch in the introduction. So I am not doing my PhD at Cambridge. I did my PhD how many? 18 years ago. I'm currently a lecturer at Cambridge at the Department of Plant Sciences. And the work that I shall be presenting to you today is essentially the way we have at Cambridge handled the challenges and the opportunities that we've had in the past 7 months, and more so in the last 4 and 5 months during the complete lockdown of the pandemic. And this work has been done at the interface of India and the UK, where my work relates to in both senses. So I'm at the Department of Plant Sciences again at the University of Cambridge, and I've been here 4 years now. I have 2 major research areas, food security and conservation. And in these 2 areas, the focus, the way I have divided my talk today is to talk to you about or present you the work that we've been doing, but not just that, essentially how we as trainers and teachers and supervisors have handled the challenges mostly that we've had during the past 6 months of lockdown, and what we've done to overcome these challenges. And during this time, the opportunities we found and the things that, and the surprises that we had on the way. Most interestingly, in terms of the students we got and the interns, especially the short-term students that we've been able to work with, and all of it online. So first of all, the research that I'm doing, first in the area of food security and genomics. So what we're doing in food security, I am a computational biologist indeed, and that is really neat because Loïc also just presented to you a lot of work that has been going on in computational biology. You will see much of that again in my talk also. We work on green carbon, and by that, what I mean is the carbon that gets fixed by Rubisco and green plants on Earth from the atmosphere. So inorganic carbon that get fixed into organic carbon through the process called photosynthesis, and which makes it extremely relevant, particularly in the times of climate change, when you need to have better ways of getting more carbon fixed and more green carbon on Earth, especially in view of the black and brown carbons that we have all around us and the carbon footprint that we are generating and expanding. So plants perform photosynthesis, and there are two major processes, as most of you would know already, but just to reiterate, it's a redox process. You have a reduction part where you have carbon fixation, and you also have an oxidation happening, which is the splitting of water via light. And this is photosynthesis, but the model organism that we're working in the lab is the guy that performs photosynthesis best, which is the organisms, the class of organisms that are able to capture carbon much better than others. And that is because they have a form of rubisco that is less, that is more efficient. And the efficiency of rubisco that is there in this class of organisms is much better than higher plants. It is because they are able to hide rubisco from the direct atmosphere, the atmosphere which has more oxygen, of course, which is why oxygen and carbon dioxide are competing for the active site of rubisco. But in algae, in unicellular green algae, carbon sequestration is far much better, often 40 times as well done as in case of crops and higher plants. And that is because they're able to hide rubisco or put it inside a little compartment called the pyrenoid, which you can see over here in the upper left corner, where we have a little drawing of Chlamydomonas. There are large numbers of data sets available in the public domain for Chlamydomonas, which we are currently studying. And these include the nuclear genome, the organellar genome, the time-resolved transcriptomes, several of them over the entire 24-hour circadian cycle, carbon-deprived transcriptomes, the organellar transcriptomes in genomes, the spatial interactomes, and so on. Our aim in all of this is to reduce the data dimensionality of the entire of all of these repositories, and to study carbon concentrating mechanism, which is the way in which CLAMI is able to concentrate carbon better than most other plants and improve its efficiency through rubisco, the enzyme that is hidden inside the pyrenoid, which is why rubisco does not see the high amounts of oxygen at all. All it sees is the carbon dioxide that is sent inside this little micro compartment. And so what we've done is we've compared all of these different data sets that we had in order to identify or get better insights into the mechanism of algal biophysical carbon concentration mechanism, but most importantly, to identify new or key components of the carbon concentration. And this is where comes in the idea of a temporal transcriptome. So we've got time-resolved gene expression data sets, very high-resolution data sets, and we've performed transcriptional gene expression, transcriptional inferences from gene expression data in order to separate the day-night cycle for CLAMI into 5 sections. We call them, based on biology, the light networks and the dark networks. The light networks themselves have been divided into the active CCM time, where during daylight from 0 to in the first 7 hours of the day, active carbon concentration or active photosynthesis is happening. Then dusk, about 2 hours, and then the division cycle. These are the 3 dark networks, or the 3 time points during the dark, where you have division, resting stage, and pre-dawn. What we have found is that the pre-dawn networks are extremely important. So what you see in this picture is the genes that we've identified from a detailed analysis that I showed you on the previous slide here. We've identified about 50 key genes, and this is where on this slide, each of the 50 key genes and their expression across the entire circadian cycle has been performed. And based on this, we do co-expression networks. So we extract co-expression data for every pair of genes, like each one of these and the rest of the 17,000 in CLAMI. And what we find is in each one of these networks, we find different parts of the CCM machinery becoming active with respect to the rest of the 17,000 genes in CLAMI. And what we found are very interesting. In these temporal gene networks, we found clues about sub-networks that get switched on or off with time. And we've been able to identify novel regulators of the carbon concentrating mechanism, especially in the pre-dawn network over here. For this, we have performed detailed co-expression network analysis based on hard thresholding as well as soft thresholding, which is WGCNA. So you see the WGCNA modules on the lower side of this this slide, but we are more interested in the hard thresholded networks because that is where we are able to quantify, actually have numbers for or pairs of genes which are correlated with each other. Currently, we are extrapolating more information about the pre-dawn networks and the regulators that we found in the pre-dawn networks. And that's the work going on in this area. The second, and this has been done by my student, my PhD student, C2, who's in her final year just now. The next part of the work is about the work we do in conservation. And that's what brings us to trying and understanding or unpacking plant chemical arsenals. This is the work that my part 2 and part 3 students at the University of Cambridge are currently performing, as well as several of my students in India. And so here I start again about ecological informatics and trying to deep learn plant chemistry. So what we've done is we've tried to look at plant emissions, and we are trying to capture plant emissions from a contextual point of view, where we want to understand how these plant emissions vary in a given species across time, across space, as well as across conditions of stress or disease and across the entire planet. Just a second, please. Yeah, I think it's working. Okay. So to measure the terpenome, what we've tried to do is we get emission data, but it's not easily possible for you. I mean, even if we wanted, you couldn't get the emission data for every plant under every circumstance. We usually do these field experiments in both in India and the UK, where we collect the headspace volatiles from plants that are growing around us and the species that we are interested in. But, you know, to be able to compare it across various habitats, across different geographical locations, you need information that is in the published domain already, because there's just no way I can do it myself or anybody else can do it, but for collecting the emission profiles of every single plant from every single place. And for that, we need the headspace volatile information and the mass spectrophotometric data available in public literature. And therefore, the idea that we have is to mine the world's research papers. Now, that's where a very major issue of copywriting comes in, which I will not go into today, but it is true that access to literature metadata is not easy. If you have a question, you're a PhD student and you want to start out in a new area, the first thing that you're expected to do, of course, is to perform a literature review. But you can't do a true literature review if you don't have access to all of the research papers that have been published in your area of research. This is where public.resource.org's president, Karl Malamud, he's in the US. He's based out in the US, but works across the planet. And he and I met up at one point at a conference, and then it turned out that he's been able to make the world's research papers available to researchers for free, to scientists for free. Karl is himself not a scientist, but he believes in making information or knowledge available to all. The only issue is you can't read those papers. So the copyright essentially stops you from reading a paper, but not your access to the literature metadata. So if I'm interested in, for example, understanding or finding out which chemicals or what chemical profiles specific plants had at different points of time from different geographic locations across the world, all I need is to extract the data that was possibly in these mass spectrometry profiles, or composition profile information that was there in these papers, maybe in a table, maybe in a figure, but I don't really need to read the entire paper as long as I can find a mechanism to scrape that paper of that information, and not just that paper, but maybe hundreds of millions of papers that have information that might be relevant to me. That is where Karl's data corpus, he made a data corpus and has made it available. If you need to read more, this was published last year only in Nature, that this repository is now available to scientists. And what we've done is we've tried to, having access to the data corpus is not enough. What we need is for students or for us to be able to scrape that information with the right kind of code. And writing that code is where our interns have come in to help us incredibly. And my colleague, Peter Murray Rust at the Department of Chemistry at the University of Cambridge, as well as Manny Faria in Brazil. He's just CEO of a company that very clear that handles a cosmetic company where he needs plant chemicals, and he's trying to build herbal medicines or herbal solutions to a lot of skin problems. And he needed constantly, he had wanted to get in touch with people who do plant chemicals or herbal analyses. When Manny and I met, I was interested in building a large corpus of plant chemistry based on published literature from the point of view of trying to understand evolution of plant chemicals. Manny came to the same question from the point of view of a cosmetologist. And Peter came to the same question from the point of view of a copyright activist. Now, Peter just doesn't like to have paywalls in the way of students and knowledge and researchers and knowledge. So the three of us came together and we built this thing together called the CEVOpen, which is, we're asking the question of whether machines can curate open phytochemistry literature and disambiguate data. And then what we went into during COVID was to put this together on OpenVirus as an online mentorship program. And this is the main thing I want to talk about today. OpenVirus is freely open on GitHub for you to see and view the progress we've made, the work we're doing, the way we're scraping literature, the kind of questions we're asking. Our lab meetings are open and live streamed. Our progress reports of each one of our interns is online. The work they're doing is online. In addition, we have a little movie that our interns have put together for us on the International Young Academy of Science YouTube channel, which you're welcome to look at. But most interestingly, this mentorship program has people from India, UK, China, everybody coming together, all of these students in their own homes voluntarily working. There's no degree you'll get out from it, but what you will learn is to write code to scrape data from literature without having to read it, without breaching any copyright laws. And I think that's extremely important. And this is where a lot of science academies have come to help us. And we've been able to build these codes together. I invite and welcome you all to look at OpenVirus. For the last 3 days, we have been at the Bioinformatics Hackathon at the B. Abraham Institute in Cambridge, talking to others about OpenVirus, about what we do, how we scrape literature. And you're more than welcome to join the community or just find out more about us by going to Twitter or to GitHub or to YouTube and checking out OpenVirus with Peter Murray Rust and myself. So back to ecology, we've been very interested in trying to understand complex networks. I talked to you about the genomic networks, and what I haven't talked to you is about the ecological networks that we've been extracting using phytochemistry for species in different habitats. And we've built up an algorithm to perform network perturbation. This was done many years ago. About 8 years ago, we built this algorithm. It's called Nextcade. And what it essentially does is it's an automated program for induction of knockouts, or in case of ecological network species, essentially knocking out species or species extinction, and assessing their impact on the remaining network in terms of secondary extinctions that might happen. For a gene or protein network, this can be extrapolated as removing or knocking out a gene and seeing what else happens. We've now improved the network perturbation algorithm earlier it was a knockout, but now we're doing a knockdown. For instance, you have a network and you want to understand what happens when the node size or the impact of a certain node reduces, rather than chucking that node out altogether from the network. And that work is now part of our algorithm development strategy. There's a lot of bioinformatics training and computational biology training that's happening at the University of Cambridge, which I want especially if we have Oxford students here, we're more than interested in having you come in and look at us and the kind of work we're doing. And if possibly we can do things together. I am an instructor at the Bioinformatics Training Facility at Cambridge, and I would like very much to connect with instructors or trainers at a similar facility at Oxford if it exists, and we'd like to do things together in Cambridge. I mean, things have changed completely from before the COVID pandemic to after. Before the pandemic, we used to have these large-scale training sessions for days and weeks, hands-on live training, teaching people, and trying to understand basic core bioinformatics and specialized training sessions also. But after the pandemic and during the pandemic, what we came up was I would never have before the pandemic agreed to a protocol from home or having to teach bioinformatics or computational biology or coding to anybody online. But now I stand corrected. It can be done, and it can be done beautifully. We've been running these online courses since April this year, and these have been extremely successful. For instance, today I spoke about the work that we've done in genomics about co-expression networks. We've run these courses entirely at the University of Cambridge as well as in India. We've been running online free courses to handle, to look at to first of all visualize your own data as a network, build biological networks, construct them and perform analyses, clustering, do co-expression network construction in various ways like hard thresholded or soft thresholded networks. and then how to, extract network motifs and so on. And then, as I just said, network perturbation. All of these tasks we've done in our research. All we have done through the Protocol from Home series is to convert these into smaller packages that students and researchers outside of our laboratories, outside of our world, may be able to learn from. Much of the, most of these talks are already online, and the course material is freely available online. I often try to use our own research data, but it's best to use published data, and therefore we've used very basic data and data sets, and we're doing these through the University of Cambridge Bioinformatics Training Facility. The other courses that are coming up now in future are again for network perturbation, some on spatial modeling and GIS, as well as in data skills. These are long-term courses where we have an introductory R on data skills for 3 days, followed by an intermediate R, and then advanced R, where we go in for plotting and clustering and single cell chip-seq analyses and stuff like that. So there is lots of capacity building that is currently going on, and I encourage you to visit the Cambridge Bioinformatics Training Facility website and register or book yourself a place on 1 or 2 of these. But if you've got, if at Oxford you have your own training system, I think it would be wonderful for Cambridge and Oxford to come together on this platform, especially during the pandemic. With that, I'd like to just come to the program that I have at the India-UK interface. It's called Tigris, and we're trying to transform India's green revolution by research and empowerment for sustainable food supplies. All of these are our Tigris partners in both countries. And through Tigris, it was when I first realized that as often as not, I think we have to talk on platforms just like Varsity Sci, where we can amplify the message that we've got. Earlier, I used to just visit schools in rural India and rural England and meet children and talk to them about the science that we're doing. But it's equally important to meet each other and tell each other what we're doing and collaborate if possible, or be able to amplify the message that we are trying to pass on, that it's not impossible to do research while you're locked down. You get locked down, but you can get up again. And that's the whole idea that I have here today. Thank you very much for listening to this. Thank you very much again for being part of Varsity Sci. I wish we would have more initiatives like this. These are my group members, my students, Charles, Yuan, Situ, Amish, Ramya, my collaborators, Howard, Allison, and others at Content Mine and Rothamsted Research in India, of course, at the department in my own college, St. Edmund's College at Cambridge, and so on. With that, I'd like to thank everyone for listening, and apologies again that I was a few minutes late getting in here. Hello? Yeah.
Thank you once again, Gita, for the very fascinating talk and on her work on promoting open science and computational biology, especially at this difficult time. So I would like to apologize for the mistake I made during the introduction.
All right. All right.
Yeah, I guess, well, this is the first time we're doing such an online thing.
I appreciate it. You should do it more often. Thank you so much for starting this out. Okay.
Thank you. Right, so now I am going to, so there's one question on Zoom. Are there any comparable data mining projects outside of?
I didn't get that, E. Can you just repeat? I heard comparable data mining projects and nothing after that.
Okay, so the question is, are there any comparable data mining projects outside of phytochemistry?
Yes, yes, there are. First of all, data mining for phytochemistry is a very small part of data mining that is happening in the universe currently. If you go back to the Nature paper that was published last year with Carl's data corpus, about Carl's data corpus, the real data mining that is happening over there is biomedical data mining and health related data mining. I am possibly one of the very few people, or my group is possibly one of the very few groups who are interested in phytochemical data mining. But in the past 6 months, now this paper was published in 2019, June, July. But in the past 6 and 7 months, especially during the pandemic, most of the data mining that has been happening across the planet is about about viral epidemics, indeed. And therefore, phytochemistry is not at all up there in the competitors. It's actually the coronavirus. And if you look at the last month's, where was this? The supercomputer at Texas, was it, or Oklahoma, where they were able to find out they were able to data mine patterns, and they were able to identify specific ways in which the virus is indeed spreading, and once it infects you, the mechanism by which it is able to have a second, if possible, more often than not, a second relapse, and so on. So there's a lot of data mining that is happening, but I think it's not in phytochemistry. And so, does that answer your question?
Yes, thank you. So there's one question from YouTube. Great talk. How would one validate that this large amount of data has been collected or mined? Would all of it need to be fact-checked by a human anyway?
Okay, that's a good question. In all text and data mining methods or protocols, what we do is we've got this data corpus. You start out with a certain data corpus. If you use protocols like AMI, which we are using in our group with Peter Murray Rust in OpenVirus, if you take a look at OpenVirus, you'll find that we're using the AMI software, to whom you can basically say you need to start out with, let's say you're just beginning. You start out with 50 papers or 100 papers, and let's say you move on to 500 or 1,000 papers. In the data corpus that we have, that we published last year with Carl, there are 73 million papers. But to begin with, if you're looking at 100 or 200 papers, you need to make sure that these 200 are indeed relevant to the question you're asking. So the manual intervention that is required is to make sure that the title and abstracts of these 100 papers are all right. But when the numbers go beyond 100 and 500, when they go into several tens of thousands, in that case, it's not possible in any way to manually do anything. So what we do is we build dictionaries. So the fact checking that is happening through dictionaries. You put certain words that are of interest. For instance, in phytochemistry, which is my bread and butter, the kind of words I'm interested in are whether certain sesquiterpenes or diterpenes or monoterpenes were released by certain plants. Let's say I'm interested in one particular species. Let's say it's just as an example, let's say it's the rhododendron ponticum, and I know very well that certain cyanotoxins are being made by this plant or this species in different places. So the kind of keywords I would put in to fact check whether those papers are indeed truly there would be rhododendron ponticum, and it would also be the presence of cyanotoxin 1, 2, and 3, and or presence of other sesquiterpenes. So there would be this list of words or terms which have to be in those papers for those papers to be not rejected or to be relevant for me. And those words or list of words of interest to you or the question that you're asking have to be in this file, which we call a dictionary. So we build dictionaries. So in the internship program, in the OpenVirus internship program, a lot of students who have really helped us incredibly is to move ahead in terms of building the right kind of dictionaries. Without dictionaries, what do you go ahead and scrape, right? So you can't just ask a question into the air and expect that the machine is going to return to you everything that you want about it. So you put these questions into dictionaries, you make it more focused, and then you scrape the data back. And then out of those 5,000 papers or 500 papers or 50,000 papers that you were looking at, whichever have the combination of these in various, some papers may have terms that you gave in your dictionary. Some may have just half of those terms or 10% of those terms, depending upon the extent to which the terms of interest are present in the terms of combinations, in the extent to which the combination of terms are present in the papers that you're looking at or not looking at, you would know whether or not this data is validated. And that is the way we do it. Does that answer your question?
Yes, certainly. So I guess if there aren't, okay, so there's one more question. Would you mind elaborating on your work with Tigris?
With Tigris, yes, indeed. As I said, in Tigris, we're trying to look at the an extended green revolution for India. So there are two ways we're addressing this. Well, there are 6 work packages and there are 6 different ways we're addressing this. I encourage you to visit the Tigris website at the University of Cambridge. But this is one of the most beautifully put together natural science and social science programs. So it's a Global Challenge Research Fund project. It's a GCRF funded BBSRC program. It's running for 4 years. It's been running for 3 years, and we have another 1.5 years to go on in this one. And what we do is we have partners in India and partners in the UK who are social scientists, economists, agriculturists, genomicists like me, computational biologists, as well as GWA specialists. The entire CGIAR system is with us. We have universities, colleges, we have women farmers, we have NGOs with us. We also have archaeologists in the University of Cambridge who are working with us, as well as botanists and physiologists at the University of Essex. And we all come together to look at various aspects of the Indian Green Revolution, which was essentially, if you remember, focusing on 2 major crops, wheat and rice. We are trying to transform that to look at the more, the more dryland-oriented species or crops like millets. And we are trying to address, as far as my work package is concerned, where we do research, but not surveys in social science, where we are interacting with farmers and other stakeholders and trying to identify the problems that they have in India. What we are trying to do in the research aspect in the second work package is to address two things mainly. One is how to enhance productivity by studying water retention better, because from an Indian point of view, the drought angle is very, very, very critical. So we're trying to find out better ways in which water is essentially split by light, as well as finding out crops that are much better at retaining water, for which we are doing a lot of GWAS work, and this is happening at ICRISAT in India. My sub part is that of understanding food security or food productivity from the point of view of better productivity. And how do you get better productivity? By performing photosynthesis better, essentially. And how do you get better photosynthesis? It's by having a more efficient Rubisco. And who's got the most efficient Rubisco? I just talked to you. It's clammy, brings us back to our beloved clammy. So we are trying to understand clammy in terms of its ability to assimilate carbon better than most other phototrophs on the planet, oxygenic phototrophs on the planet. I encourage you to look at the website. Indeed, it's a lot to take in one go.
Thank you once again for answering those questions. One interesting thing is that we just had one plant science talk, and the speaker was really fascinated about pyrenoids and the concentrating mechanism. So yeah, it's interesting how these talks tie together. So thank you once again. I'll now pass the floor to Christoph to wrap up the session.
Thank you.
So thank you so much, Gita and Loïc, once again for those 2 brilliant talks. Thank you to Loïc for encouraging us to think about computation and the impact of computation in a different way, thinking about environmental costs. And I think that's also very relevant to a topic that I believe is quite current at the moment in computer science, thinking about optimization of algorithms and about how some of that has been lost as hardware improved thanks to Moore's law and to technological developments. And thank you to Gita for introducing us to her many initiatives, both in India and the UK, which share a common theme of open data, open science, and communication, which I think is a topic very, very much at the heart of Varsity Sci, and appropriately enough links in with topics that we've also discussed in other sessions. So for example, if you'd like to find out more about pyrenoids, check out Ella's plant science session, which was published in the evening yesterday. And I believe that open data in the context of the current pandemic is something that our keynote speaker on Saturday, Dr. Apweiler, will discuss in more detail, as he's the European coordinator of a project that aims to do exactly that. So once again, thank you very much to everyone who joined us, to our 2 brilliant speakers. And we hope to see you soon at 1:45 actually, for our first physics session of Varsity Sci. So thank you once again, everyone.